Hello everyone, we're going to start today the first lesson in biology for grade 9. Okay, so the first chapter that we're going to talk about is enzymes. What do we know about enzymes? Enzymes here are said to be proteins in nature, so enzymes are made from proteins. Okay, so these proteins or these enzymes, they actually functionize as biological catalysts. Then what is a catalyst? The catalyst here is said to be that it's a substance that we use in chemical reactions in order to speed it up. For an example, I had two substances. I want them to react together, but this reaction is going to take up to, let's say, 1 hour, for an example. But I don't have time to stay until 1 hour to see what sort of product is going to be there. So what do? I would add a catalyst, which is a substance that speeds up the chemical reaction. Instead of having it during 1 hour, I would have it let's say half an hour. So practically it's rushing the chemical reaction without getting changed. The catalyst does not get changed by the reaction. Okay, so the catalyst here is a substance that speeds up a chemical reaction and it's not changed by the reaction. And the enzymes in general are proteins that functionize as biological catalyst. The enzymes here lowers the amount of energy needed for the reaction to take place and the activation energy needed for the reaction to take place. And also there's a lock and a key theory regarding how the enzymes actually work. Okay, imagine that you have got a key and the keyhole. The whole harmony and how the key suddenly fits into the keyhole. This is what we are trying to explain here as how the enzyme makes the enzyme substrate complex. As you can see here, this is the shape or the morphology of the enzyme. Focus here on how the shape has its own sort of figure. And here, the substrate. Each enzyme in our body would actually bind to a substrate. But in order to be the whole binding process to succeed, the substrate has to have the exact sort of fitting to the enzyme shape. For an example, if we had it here half spherical, or we have it as a square shaped, will it still be fitting in the enzyme? No. So practically no enzyme substrate complex will be formed. But the enzyme always looks for the substrate that exactly fits in. And that's why they named it the lock and the key theory, because the enzyme or the substrate fits exactly to the enzyme. The site where both are fitting, it's called the active site. So when I ask you where is the active site for the enzyme and substrate complex, you will say it's over here. It's where the enzyme is actually bounded to the substrate. After we have the whole fitting thing, we will call it now the enzyme substrate complex. The substrate here is the molecules before they are made to react. Okay, in any chemical reaction, these are called reactants or enzyme and substrate, and afterwards, after they bound, we will call them a product. Like for an example, when we took in chemistry, if I add any sort of, let's say, oxygen with hydrogen, both of them are actually reactants. But after they binded, what do we call whatever the water that has formed? We will call it a product. So a product is actually what has been made from two substances are mixed together. The catabolic reactions are molecules which are breaking down. Meanwhile, the anabolic reactions are molecules that are combined. As any sort of of reaction, there are so many factors that affect the activity of the enzyme. Okay, one of the things that actually affects the enzyme is we've got temperature, we've got the pH, and we've got the concentration of the substrate. So for an example, if I'm going to talk about the temperature, I would certainly go for this simple example. For an example, I have, let's say, I have this car. If I want to turn it on and like the temperature is below, let's say 50, below 50. If it's below 50, would it actually like turn on? And like I've been told that the perfect temperature for it to work on or the engine to be turned on, it's around 65. And if it's below 60 or below 50, for an example, it will be like turning on very slowly that I can't actually drive it. But if it's around, the temperature was around 65, it would be the best temperature for me to actually like turn on the car and drive. And if it's more than 65, let's say 70 or 80, it will actually turn to something else, and the engine sometimes like goes through a corruption and error and it won't turn on the car. The same thing happens to the enzymes. The enzymes here work at a temperature, specific temperature, which is called the optimum temperature, which is usually around 37 degrees. If it's below the 37 degree, here it will be like the enzymes will go through a slow sort of activity. But if it's more than 38, 39 or 40, or let's say even up to 45, here the enzyme will go through a process which we call denaturation. What is the meaning of the term denaturation? Denaturation here, for an example, as we had here, that the enzyme has this specific shape in order for the substrate to bind with it. But when the temperature is more than the usual, this whole shape will actually change to another shape, and the substrate will no longer be able to bind to the enzyme. So what do I understand from here? I would understand that the effect of temperature on enzymes, that enzymes have an optimum temperature, the temperature at which they work best and giving the fastest reaction is equally or almost 37 Celsius degree. When the temperature increases, the molecules move faster, so they collide with the enzyme in less time. Having more energy makes them more likely to bind to active site. But if the temperature is too high, the enzyme molecules vibrate too vigorously and enzymes is denatured. That means it loses its shape and no longer binds with the substrate. When the temperature is too low, there is not enough kinetic energy for the reaction, so it will be so slowly. As for the effect of the pH on the enzymes, enzymes are so sensitive to the pH, and some enzymes work best in acid than others in alkali. The enzymes here work best at their optimum pH. If the pH is changed, then the enzyme will be denatured and no longer fit the substrate, so no reaction takes place. Enzymes and their uses. Okay, we use the enzymes here like as a form of seeds to germinate. The enzymes turn the insoluble food stores to soluble. And the biological washing powders, like those what we use like nowadays with the washing machines, you will have those tiny colored particles mixed with the washing powder. What is the function of that? The function of this is that it actually removes the stain, and each one of them is actually specified for some sort of like kind of a stain. For an example, we've got the lipase enzyme, which we use it to remove the lipids like fat and oil and greasy fingerprints from the fabric. Protease is responsible for removing proteins from blood stains. As for food industry, isomerase converts the glucose to fructose, which is a type of sugar, which is sweeter, so less is needed to give a sweet taste. And pectinase, which is practically an enzyme that helps to break down the cell walls in fruit juice. For an example, I've got a lot of apples or a lot of oranges, and when I just squeeze them, like a part of the juice gets out, but I'm pretty much sure there is a lot of juice trapped inside of the cells inside of this fruit. So what do? I could actually here add pectinase to it to help it break down the cell walls in the fruit juice production, so it increases the yield and lowers the viscosity and reduces the cloudiness, and by here I would have much more of the juice. And thank you for listening.